Welcome to my presentation on the stages of mitosis. So what is mitosis? Mitosis is the process by which two genetically identical daughter cells are produced that are also identical to the parent cell and are diploid in the nucleus. Mitosis is essential for growth and repair of cells. Mitosis is preceded by a stage known as interphase, where the cell isn't dividing. There are three main substages within interphase. G1 of interphase is basically where the cell will grow and start producing the proteins that may be needed later in the cell cycle. S1 of interphase is where the cell undergoes semi-conservative cell DNA replication, and this is done in order to ensure that there is enough DNA for the two daughter cells that are to be produced later on. G2 of interphase is essentially where the cell will grow in subcellular structure and duplicate the organelles that it contains. The first major stage of mitosis is known as prophase. Within prophase, the chromosomes will condense, getting shorter and fatter, thus allowing them to become visible, whilst the nucleolus then begins to break down and disappear. The nuclear envelope then breaks down as well, also causing the chromosomes to lie free in the cytoplasm. The centrioles then will move to opposite poles of the cell, and the spindle apparatus will slowly begin to form. Metaphase is the second stage of the cell cycle. In metaphase, the two chromatids are joined together by a centromere, which make up a chromosome. The spindle network has a chromosome attached to it via the centromere. Next, what will happen is the chromosomes will then begin to move towards the equator of the cell as they are pulled by the spindle network. Anaphase is the third stage of mitosis. In anaphase, the centromere will divide into two, thus causing sister chromatids to move to opposite poles of the cell upon the spindle apparatus contracting. The energy used to drive the spindle apparatus contraction is provided by the mitochondria around the centrioles, which provide the molecule known as ATP. The final stages of the mitotic cycle include telophase and cytokinesis. In telophase, at each of the respective poles, the nucleuses will reform, as at each of the poles will have a cluster of lots of chromosomes. As a result, here the chromosomes will then unwind, causing them to eventually be unseen. Telophase is followed then by cytokinesis, where the cell membrane and organelles and cytoplasm will therefore divide again to produce two genetically identical daughter cells that are diploid in the nucleus. Cancer, on the other hand, basically will arise due to damage in our genes that control cell division. This can result in uncontrollable cell division, resulting in the accumulation of a large number of abnormal cells, which is known as a tumor. A benign tumor is one that grows relatively slowly and isn't attacking and destroying surrounding cells. On the other hand, a benign tumor can become malignant if it begins to attack and destroy surrounding cells, which is also known as cancer. What drugs, therefore, can be used to treat cancer? The treatment of cancer involves the use of drugs that will partially block an element of the cell cycle, therefore causing the cells to undergo apoptosis or G0. An example may be preventing DNA replication in S1 of interphase, or the drugs may interfere in metaphase, therefore preventing the complete formation of the spindle apparatus, and this prevents the spindle apparatus from separating the sister chromatids. Therefore, two new nucleuses cannot reform because the chromosomes won't separate. The drugs will often target rapidly dividing cells, thus causing healthy cells such as hair cells to also die as a result, alongside the cancer cells, which is why one of the side effects of cancer treatment is often hair loss. Thank you for watching my presentation. If you found this useful, please remember to subscribe. Thank you.